Welcome to The Explainer. We talk a lot about the fat we get from our food, but have you ever thought about how our body is also its own master builder, running an incredible internal factory? Well, today we're going to pull back the curtain on the amazing molecular machinery that constructs essential molecules like fatty acids and cholesterol, all starting from the simplest little two-carbon building block. So here's our game plan. We'll kick things off with the basic raw materials. Then we'll take a tour of the assembly line to see exactly how a fatty acid gets built, step by step. After that, we'll check out the critical on-off switches that control the whole operation, and see how the final products can be customized. And to wrap it all up, we'll see how that same process gets cleverly adapted to build one of biology's most famous molecules, cholesterol. All right, our story begins in the cytosol. You can think of it as the main workshop of the cell, that sort of jelly-like stuff that fills everything up. It's right here that the entire process of building a fatty acid gets started, and it all boils down to two key molecules that are primary raw materials.
Now, before we go any further, it's absolutely essential to get this one thing straight. The cell is a master organizer. To prevent total chaos, it keeps its construction sites and its demolition sites in completely separate locations. So fatty acid synthesis, that's the building process, happens out here in the main workshop, the cytosol. But fat breakdown, or oxidation, is walled off inside the cell's power plants, the mitochondria. Location, location, location.
Okay, let's meet our two main players. First up, we've got acetyl-CoA. It's the standard, go-to two-carbon building block for tons of things in the body. But for making fatty acids, the real star of the show is this other molecule, malonyl-CoA. And if you look closely, you can see it has an extra carbon group attached, making it a three-carbon unit. This isn't just a bigger piece, it's an activated, high-energy building block, totally primed and ready for construction. So that leads us to the first big question, right? The very first committed step in making a fatty acid is turning that basic two-carbon acetyl-CoA into the supercharged three-carbon malonyl-CoA. So how in the world does the cell pull off that transformation? Well, the answer is this incredible piece of molecular machinery called acetyl-CoA carboxylase. It's not just a simple enzyme, it's more like a sophisticated two-part factory. It's got two separate workstations, the biotin carboxylase site and the trans carboxylase site, and they're connected by a nifty little mobile arm. So here's how this little factory line works. In step 1, at the first workstation, the enzyme burns an ATP molecule. It uses that energy to load a single CO2 molecule onto a little hook, a cofactor called biotin. Then, in step 2, that flexible arm physically swings that CO2 all the way over to the second workstation, where it slaps it right onto an acetyl-CoA molecule. And voila! This is the net result of that whole process. We've used a little bicarbonate, we've invested 1 ATP for energy, and we've successfully added that carboxyl group to acetyl-CoA, creating our activated building block malonyl-CoA. And this reaction, this is the point of no return. Once malonyl-CoA is made, the cell is officially all in on making a fatty acid. All right, let's get to the main event. Now that we've created our activated building block, it's time to see how these units are put together on this amazing molecular machine called fatty acid synthase. What's really fascinating is how this whole assembly line evolved. In bacteria, it's like having 7 separate workers, each with their own tool, doing their part of the job. But in vertebrates like us, evolution decided to be more efficient. It fused the genes for all 7 of those functions into one single giant multi-tool protein. It's way faster and much more controlled. At the very heart of this giant enzyme is a crucial component called the acyl carrier protein, or ACP. It's got this long, flexible arm that acts like a mobile crane. It grabs the growing fatty acid chain and literally swings it from one workstation to the next, all within this single massive protein. And at the very tip of that arm is a reactive group that does all the work. So building a full fatty acid chain is actually a repeating cycle of 7 different reactions. And each time this cycle turns, it adds 2 more carbons to the growing chain. Let's walk through one full cycle right now and see exactly how this molecular assembly line does its thing. First things first, you gotta prime the machine. So in steps 1 and 2, our 2 starting materials get loaded onto their designated spots. A basic 2-carbon acetyl group gets attached to a little holding bay. Then, our activated 3-carbon malonyl group gets loaded onto that swinging crane arm of the ACP. And here is where the genius of the design really shines. In step 3, the 2 pieces come together. And as they join, that very same CO2 molecule we spent energy to add earlier gets released. This release of CO2 is what provides the energy, the driving force, that powers the whole reaction forward.
creating a new 4-carbon chain that's now attached to the ACP arm. It's brilliant.
Okay, the new chain is made, but it needs a little bit of chemical processing. In step 4, we have the first of 2 reduction reactions. The enzyme uses a helper molecule called NADPH to change a reactive keto group on the chain into a more stable alcohol, or hydroxyl group. Next, that ACP arm swings the chain over to the next workstation. In step 5, an enzyme does exactly what its name implies. It dehydrates the chain, pulling out a molecule of water. And removing that water creates a carbon-carbon double bond. We're almost done with this first cycle. The arm swings again, and for step 6, a second NADPH molecule comes in. Its job is to get rid of that double bond we just made. So what we're left with is a fully saturated, nice and stable 4-carbon chain, still hanging on to the ACP. And finally, we have step 7, translocation. In the last step of the cycle, the completed 4-carbon chain gets moved off the ACP's swinging arm and transferred back to that original holding bay. This clears the crane, so to speak, getting it ready to pick up a new building block for the next round. And from here, it's basically rinse and repeat. The entire 7-step cycle happens all over again. The new 4-carbon chain now acts as the starting piece, and it gets combined with another malonyl-CoA to make a 6-carbon chain. This whole loop runs 6 more times, adding 2 carbons each round until a 16-carbon chain called palmitate is finally built. Then, a final enzyme comes along and just snips the finished product off the assembly line. Now, with such a powerful and energy-hungry assembly line, the cell can't just let it run wild. That would be a huge waste. So it needs some pretty sophisticated control systems to turn it on when it's time to store fat and turn it right back off when it's not. Let's see how this thing is regulated. The main control knob is on that very first enzyme we talked about, acetyl-CoA carboxylase. It gets controlled in 2 main ways. First, you have allosteric control, which is kind of like a dimmer switch. A molecule called citrate, which basically signals we have lots of energy, turns the production up. The final product, palmitoyl-CoA, provides feedback saying, okay, we have enough, and turns it down. The second method is like a hard on-off switch. The hormone insulin removes a phosphate group, flipping the switch on. Other signals, like the hormone glucagon, add a phosphate, flipping it off. This gives you the big picture of how our hormones control all of this. After you eat a meal, your blood glucose goes up, which triggers insulin. Insulin's job is to tell the cell to store that extra energy, so it flips the switch to on for making fat. On the other hand, when blood glucose is low, the hormone glucagon comes out. It flips the switch to off, shutting down synthesis so the body can burn the fat it already has for fuel. But beyond these immediate controls, the cell also thinks long-term by changing how many of these factories it even builds. Insulin doesn't just activate the existing enzymes, it also tells the cell's nucleus to start making the blueprints to build more of them. And other signals, like the hormone leptin, which comes from our fat stores, do the exact opposite, telling the nucleus to slow down production. You know, that 16-carbon palmitate chain that comes off the main assembly line is really just the beginning. It's like the standard model. But now, cells can take that product and modify it in all sorts of ways to create a huge variety of specialized fatty acids with different jobs and different properties. So the cell has 2 primary customization tools it can use. First, there are elongation systems, which use enzymes to add more 2-carbon units, making the chains longer than the standard 16 carbons. And second, there are desaturases. These are enzymes that introduce double bonds into the chains, turning those saturated fats into unsaturated ones. Now, this is where things get really fascinating. One of these customized fatty acids, a 20-carbon molecule called arachidonate, is the starting point for some incredibly powerful signaling molecules. When a cell gets damaged, an enzyme snips arachidonate out of the cell membrane. Then, an enzyme called cyclooxygenase, or COX, gets to work on it, starting a cascade that produces things called prostaglandins, the chemical messengers that trigger pain, fever, and inflammation. And that right there explains how some of the most common medicines on the planet actually work. NSAIDs, or nonsteroidal anti-inflammatory drugs like aspirin and ibuprofen, they work by going straight for that COX enzyme. They physically block it, stopping this entire pathway in its tracks, which prevents the production of the prostaglandins that cause all that inflammation and pain. All right, let's move into our final section. We're going to see how the cell uses the exact same starting material we've been talking about, acetyl-CoA, to build a completely different lipid superstar, the famous, complex, and absolutely essential molecule cholesterol. And here it is in all its glory, cholesterol.
It's a 27-carbon behemoth with that distinctive 4-ring core. It looks incredibly complicated, and it is. Its synthesis is a true masterpiece. But here's the mind-blowing part. Every single one of its 27 carbon atoms originates from that simple 2-carbon acetyl-CoA molecule. So this journey starts by taking 3 of those acetyl-CoA molecules and stitching them together. That creates a really important 6-carbon intermediate called HMG-CoA. This might sound a little familiar, actually, because this same molecule is also used in making ketone bodies. But the very next reaction, this is the crucial one. This is the point of no return for cholesterol. An enzyme called HMG-CoA reductase converts HMG-CoA into another molecule, mevalonate. This reaction is the committed rate-limiting step. Just like its counterpart in fatty acid synthesis, this one enzyme is the master control switch for the entire cholesterol pathway. Okay, so once mevalonate is formed, the cell invests even more energy to process it into these activated 5-carbon units called isoprenes. You can think of these as the fundamental Lego bricks of cholesterol. The rest of the long, complicated synthesis pathway is basically just about linking these little 5-carbon bricks together in various combinations to eventually build up that full 27-carbon structure.
And that brings us directly to statins, one of the most widely prescribed classes of drugs in the world. How they work is just beautifully simple. They are designed to look almost exactly like HMG-CoA, so they fit perfectly into the reductase enzyme and just jam it up. By shutting down that one single master switch, statins effectively turn down the dial on the body's entire rate of cholesterol production. So, we've seen the incredible power of our internal factories to build these complex and vital lipids from the simplest of starting materials. And that leaves us with a final, huge question that's really central to our health. How does the body balance this internal manufacturing against the fats and cholesterol we get from our diet? Understanding that delicate, complex balancing act, well, that's one of the great challenges of modern medicine and nutrition.